Hello and welcome. Now we are going to talk about one of the most important file formats in sequencing data analysis, the FASTQ file. If you work with next generation sequencing or NGS, this is the very first file you will encounter. So understanding FASTQ is absolutely essential. Let's start with a simple question. What is a FASTQ file? A FASTQ file is a text-based format that stores sequencing reads, including the DNA sequence, a unique identifier for each read, and the quality score for each base. In simple terms, it tells you what was sequenced and how confident the machine is about each base. Now let's understand the structure. Each read or entry in a FASTQ file consists of 4 lines. Let's look at an example. Line 1 starts with a @ sign or symbol. This is the identifier or header, which is a unique label for each read. In Illumina sequencing, this line is not random. Rather, it follows a well-defined structured format. The standard header format used in Illumina sequencing is shown here. As mentioned earlier, the header starts with an @ symbol or sign, followed by instrument ID, run number, flow cell ID, lane number, tile number, X coordinate of the cluster, Y coordinate of the cluster, and optionally a UMI or unique molecular identifier. All fields in this part is separated by clones, and this portion is referred to as the coordinate section. After this section, there is exactly one space followed by the read information section containing the read number, filter status, control number, and index sequence. All fields in the information section are also separated by clones. Note that there is exactly one space separating the coordinate with or without the optional unique molecular identifier field and the read information section, and no additional spaces are present in the header. The read number field contains either 1 or 2, where 1 represents read 1 and 2 represents read 2 in paired end sequencing. The filter status field contains either Y or N, where Y indicates that the read failed quality filtering, while N indicates that the read passed the filter. Here is the standard header format without the optional unique molecular identifier or UMI field. As we can see here, unique molecular identifier field is absent here. However, the coordinate section and the read information section are still separated by exactly one space. Here is the example of standard header format without the optional unique molecular identifier or UMI field. Now let's see the precise breakdown of the identifier or header used in this example. It starts with a @ sign or symbol, which marks the beginning of a FASTQ entry. Next comes the instrument ID, which is SIM here. Then the run number, which is 1. After that, the flow cell ID, FCX. Then comes the lane number, lane 1. Next is the tile number, tile 15. These two numbers represent the cluster position on the flow cell. The X coordinate of the cluster is 6329. The Y coordinate of the cluster is 1045. Next we have UMI sequence. As we can see here, UMI stands for unique molecular identifier. After the space, we move to the second section of the identifier. Here we have the read number. As we can see here, 1 is the read number. 1 means this is read 1. If it were a paired end sequencing, read 2 would be marked as 2. Next is filter flag. N means the read passed the quality filtering. If it were Y, it would mean the read failed filtering. Then comes the control number. Here it is 0, meaning no special control was used. Finally, we have the sample index sequence. As we can see here, this index helps identify which sample the read belongs to during multiplexed sequencing. Line 2 in a FASTQ entry is the sequence. This is the actual DNA sequence composed of the nucleotide bases A, T, G and C. As we can see here, line 3 is a separator line. This is also called quality score identifier line. In older FASTQ formats, this line could repeat the identifier or header information. However, in most modern FASTQ files, it usually contains only a plus symbol. Line 4 contains the quality scores. This line represents the sequencing quality of each base in the sequence. As we can see here, a very important point to remember, each base in the sequence has exactly one corresponding quality score in the quality line. So if the sequence contains 100 bases, the quality line must also contain exactly 100 quality characters. A FASTQ file contains thousands to millions of these 4-line entries repeated one after another. That's why FASTQ files often become very large. Each entry in a FASTQ file represents a single sequencing read, and each read is generated from an individual cluster on the flow cell in Illumina sequencing. Now comes the important part. What are these quality scores? The quality score of a base, also known as a Fred score or Q score, is an integer value representing the estimated probability of an error. That is, the probability that the base is incorrect. In other words, it tells us how likely it is that a base call contains an error. These quality scores are based on Fred quality score system. So the quality score reflects the probability of error in base calling. The formula for calculating the Q score is shown here. Here Q is the quality score and P error is the probability that the base is called incorrectly. Based on this relationship, as we can see from the table, Q20 means 1% error, Q30 means 0.1% error, Q40 means 0.01% error. So higher quality score means lower probability of error and therefore higher confidence in the base call, as we have discussed in the previous lecture. Now let's go deeper into something very important. How exactly are quality scores stored as characters? First, let's see what is ASCII encoding. FASTQ does not store quality as numbers. Instead, it stores them as characters using ASCII encoding. Storing quality scores as ASCII characters is more compact than writing numbers like 30, 33, 35, 40 because each value is stored as a single character. Here we can see these are the ASCII characters in the fourth line of a FASTQ entry. ASCII is a standard encoding system where each character such as capital A, capital B or exclamation mark is assigned a numeric value or code. In this table, these columns represent the ASCII characters and the decimal columns show the numeric value or code corresponding to each character. Each quality character in FASTQ corresponds to a Fred score plus an offset. So the stored value is not the direct quality score. Instead, it is encoded like this: character equals quality score plus an offset. In other words, the ASCII character represents the quality score plus an offset value. Since each ASCII character corresponds to a numeric ASCII value, we can get the actual quality score using a simple formula: Q equals ASCII value minus offset. We will discuss the offset in details later. Now, what is Fred plus 33? In modern sequencing, we use Fred plus 33. In this system, the Fred quality score is converted into a ASCII character by adding an offset of 33. For example, Q0 corresponds to an ASCII value of 33, which is represented by the character exclamation mark. Similarly, Q30 corresponds to an ASCII value of 63, which is represented by the character question mark. While Q40 corresponds to an ASCII value of 73, which is represented by the character capital I. So when you see the character capital I in the quality line of a FASTQ file, it represents a very high quality base call with a Q score of 40. Now let's decode the first two quality score symbols from the FASTQ entry we saw earlier. As we can see here, first two quality score symbols are less than and greater than symbols. For the first base, the character less than symbol has an ASCII value of 60. To calculate the Fred quality score, we subtract 33. This gives a Q score of 27, which indicates moderate to high confidence but not extremely high. For the second base, now let's look at the second character, the greater than symbol, which has an ASCII value of 62. Applying the same formula, this gives a Q score of 29, indicating slightly higher confidence than the first base. Older sequencing systems used Fred plus 64, but this is now obsolete and only seen in older datasets. Now an important question: why do we use an offset? As FASTQ is a text format that must store one character per base, that is, fixed width is needed, and it must avoid non-printable characters that can break the file structure. As we can see from the table, ASCII values from 0 to 31 and 127 are non-printable, while 32 to 126 are printable characters. Although a space is technically a printable character, however, FASTQ does not use spaces in quality strings to maintain strict formatting and avoid ambiguity. So what are non-printable characters? These are characters that you cannot see them on the screen as letters, numbers or symbols. Non-printable characters are characters that don't display visually, they control formatting instead of showing symbols. These characters are useful for formatting text but not for storing visible sequencing data. For example, ASCII value 0 is null, which represents no character or an empty value. ASCII value 7 is bell, which triggers a beep sound in some systems. ASCII value 8 is backspace, which deletes the previous character. ASCII value 10 is line feed or new line, which moves the cursor to the next line. ASCII value 13 is a carry as return, which moves the cursor to the start of the line. ASCII value 27 is escape, which is used for escape or control commands. So now, why this matters in FASTQ? If FASTQ used these values directly, quality values would become invisible and the files would break. So we add an offset to shift all the values into printable characters. So if we stored quality directly without using offset, Q equals 0 would map to a ASCII value of 0 as we can see here, which is a non-printable invisible character that is null. That would break the FASTQ file. Now imagine if a quality character becomes a line feed or new line character, the FASTQ structure or format would immediately break. For example, this is a correct FASTQ entry with 4 lines as we can see here. Line 1 is sequence ID, line 2 is sequence, line 3 is separator, line 4 is Fred quality scores. But if a new line appears inside the quality line or string, for example at the third base, the structure would break like this as we can see here. After the second base, there is a new line character. Now the entry becomes 5 lines instead of 4. This completely breaks the FASTQ structure or format, and downstream analysis tools can no longer parse this file correctly. This is exactly why FASTQ avoids non-printable characters and uses offset encoding to ensure all characters remain visible and safe. Now let's move on a step further. Not all FASTQ files are stored in the same way. Depending on the sequencing method and storage format, FASTQ files can exist in different types or format. First is single-end FASTQ. This is the simplest format. Single-end files contain one read per fragment. So we get one FASTQ file. This file contains all the read in a single direction. Next is paired end FASTQ. This is the most common format, especially in Illumina sequencing. In many sequencing experiments, we generate paired end reads. That is, each DNA fragment is sequenced from both ends. So we get 2 separate FASTQ files such as sample_R1 FASTQ, sample_R2 FASTQ. Here the first file is read1 or R1 or forward read file, and the second file is read2 R2 or reverse read file. The IDs for both reads are almost the same in paired end reads. The only difference is the read number in the header line. As we can see, for read 1 it is 1 and for read 2 it is 2. Now instead of storing read 1 and read 2 in separate files, some pipelines store them in a single file. This is called interleaved FASTQ. As we can see here, reads appear in pairs one after another. As we can see, this is read 1 and this is read 2. The key idea is that read 1 and read 2 are alternating in the same file. Then the compressed FASTQ files. FASTQ files are usually very large because sequencing generates millions of reads. So to reduce file size and make data transfer easier, they are often stored in compressed format. As we can see here, the most widely used format is .fastq.gz. As we can see here, you may also see fq.gz, which is just a shorter form, and occasionally .fastq.bz2, which uses a different compression method but is less common. The important point is, even though the file is compressed, the internal structure is still FASTQ format. Most tools like FastQC, BWA and BOT-I2 can read compressed FASTQ files directly. So to summarize, FASTQ files contain millions of reads. Each read follows a strict 4-line structure. They store both sequence and quality information. Quality scores are encoded using ASCII. Offset ensures all characters remain printable, and maintaining the structure is critical for correct analysis. FASTQ files exist in different formats such as compressed files and paired data structure. So now you fully understand FASTQ files from structure to encoding to real-world implication and why it is so important in NGS analysis. This FASTQ file forms the foundation and marks the start of NGS data analysis. That's all for now. Thank you everyone.